When we talk about microphysiological systems, what we're talking about is a way of growing human cells in the laboratory in such a way that they don't just grow in a flat sheet, but they actually develop the lovely hilli and grooves and shapes that exist inside your body. And this is a massive advantage in terms of growing viruses which need that type of differentiated tissue. We're now able to do that using these miniature systems which we can interrogate with some very sophisticated tools.
So ultimately what we're trying to do with these human organ chip models is to have models of virus infections in a human system so that we can test countermeasures such as antivirals or vaccines. So when the next pandemic comes along, we're just ready to go. So we obtain human cells either from commercial sources or from human biopsies. And when they come into the cell, we culture them in a way that encourages them to differentiate into the cells, into the structures that we see in the human body. This is through a variety of chemicals and growth factors and growing them at a temperature at which you would expect in the human body. When we infect our organ chip systems, we can look for various different readouts of virus infection. So this can be as simple as looking down a microscope and looking at the effect that virus has on the cells. If it's killing the cells, that's really quite obvious down a microscope. But there are other readouts, such as looking at the amount of virus that is being produced by the cells. We can look at the immune response, and we can also fix the tissue and stain them in a way that is already done in vivo studies, and we can see the same kind of pathology as we see in those tissue samples.
Traditionally, we've had to rely on animal models, and that has proven a very successful pathway. But the future does look like we will be able to use MPS in the future to screen drugs and give us that assurance that they work against humans.